Perfect. Thank you. Yeah. So first off, to explain the name a little bit, YACHT stands for Yes, No Answers to Community Membership via Hypothesis Testing. So this is going to be a methods talk, basically. And so I'm going to motivate this particular method, talk about the algorithm itself, a few experiments, and then conclude at the very end and share the code. So first off, with the motivation, what is the actual problem that we're trying to solve here? So basic idea is that we have a whole bunch of raw metagenomic reads. And then along with that, we also have a database of microbial genomes, or just genomes, period. And one of the most fundamental tasks is to be able to answer the question, are there any, and I can get my laser pointer here, are any of these reference genomes represented in my sample or in my reads? And so what we'd like to do is be able to go one by one at a very, like high level of abstracting this and be able to say that amongst these about 1 million different genomes, this one is present, this one is absent, et cetera, et cetera. And this is kind of, you can think of as a proto step before taxonomic profiling, which is also getting relative abundance. So we want to get a vector of 0 and 1s saying presence and absence of these genomes in my sample. But there's a very difficult problem, which is disentangling the noise from the rare microbiome that was actually mentioned in the previous talk. So if you look at a plot that looks like this, where on the x-axis we have the ordered taxa or protein or whatever it is that you're trying to classify in your metagenome, and the relative abundance on the y-axis. A lot of the computational tools that are out there have this long tail of noise, and that unfortunately is exactly where the rare microbiome sits. So disentangling between low abundant organisms and ones that are noise because of whatever profiler tool that you're using is a difficult task. Okay, so what do people do in practice? Well, hard thresholding is one of the main things. People just stick a threshold and say, well, we're just going to throw away everything below this relative abundance. We might be throwing out some true positives, but that's probably where all the noise is. So we'll just set a threshold there. The unfortunate thing is that in practice, you lose a lot of that rare microbiome where a lot of the interesting stuff can happen. So for example, this is on a simulated data set, Bracken, one of the popular taxonomic profilers, and the ground truth, because this is a simulation. And you can see that, yes, indeed, this tool has a long tail of low abundance noise, but unfortunately the low microbiome here, or the rare microbiome, has low abundance. And so if you're to set an arbitrary threshold, you'd be losing that interesting piece. This happens pretty universally, save for marker gene approaches. So you heard a bit about Metaflan earlier, marker gene approach. Well, in the Chemi benchmarking study, we've seen that pretty much all the profiling tools suffer this noise in that low abundance tail, save for the marker genes, Mo2s, and Metaflan, which trade off that for sensitivity. So they don't have that long tail, but they have a little bit reduced sensitivity. Okay, so what is YACHT? Well, I'm going to be talking about ANI, and there's actually ANI mentioned in the previous talk as well. So ANI, the average nucleotide identity. So basically, if you take two strings, you align them up, you take the regions that align together, and you look at the ratio of the matching and all nucleotides, so matching and mismatching. So here, for example, I have two strings of length 15, and highlighted in red here are a couple of SNPs or mismatches between them. And so the ANI between these sequences would be 13/15. As mentioned previously, ANI is a great way to be able to talk about the relatedness of microbes. And so you can have these nice rules of thumb that say like, oh, 95% ANI or higher, these are probably the same species. 99.5% or higher, these are probably some sort of strain or same subspecies, etc. And it also goes in the other direction if you want to talk about higher taxonomic levels, genus, etc. 80% or less similar. Okay. So before talking about actual YACHT, a couple of observations that were actually made in the previous talk as well. So thank you for doing some of the prep work. No organism in your reference database is going to exactly match something in your sample. Because of the strain variation, you are not going to find an exact match basically ever of something in your reference database. And so saying one of these, the problem that I set out to talk about in the very beginning, the presence absence, is actually ill-defined. Everything is absent because you're not finding the exact same representative. However, they might have a high ANI or a high relatedness to something that's in the sample. So for example, if you have these couple of microbe reference genomes over here and have ANI 0.97 and 0.92. So you might be able to say, oh, well, something of the same species as this reference genome 1 is represented in my sample, but maybe not the same species as this genome 2, because maybe my threshold was set for the ANI to require higher relatedness. The other thing is, since we're talking about the rare microbiome, you aren't going to get 100% coverage. So if you take in the black here, this is a reference genome, and you look at all the reads that have sequenced it, oftentimes, if it's a low abundance organism, you're going to have less than 1x coverage. So what we would like is to have some sort of statistical test to tell us if a genome is present or absent to my sample, and critically, based off of ANI. So you're never going to say this exact genome is present, but you can say something at this ANI or higher is present in your sample. And this is what YACHT does. Okay, well, if you're given a confidence value, say 95%, I want to be 95% sure that you fix a desired false negative rate. So we don't want to be missing out on things. You set as a user what percent ANI you say means same genome. And if you're talking about just at species level, maybe that's 95%. If you're talking subspecies, maybe set it at something a little bit higher. If you're like, yeah, whatever, genus is fine, you can set your ANI lower. You set what percent of the unique regions to the organism have to be covered by reads to be represented as in your sample. After that point, YACHT will go and tell you if there is enough evidence in the form of overlapping k-mers to support the reference genome being in or out of your sample. So no more of this hard thresholding and ignoring things. Instead, we're going to go organism by organism on the reference genome and ask, do I have enough evidence to say that an organism is in the sample that's related to this reference genome at ANI 95 or above, or 96 or above, or whatever you want. So this is an algorithm, and I'm definitely not going to go through the details. I just want to give you a flavor of kind of what is going on here. Basically, the heart of it is just a little statistical test right here that I'll explain in a minute. But why have we not had a test like this beforehand in metagenomics? Because actually there were just 2 recent developments that have allowed this sort of test to work at scale. Those 2 recent developments are, one, relating k-mers, which, by the way, k-mer just a string of length k. You chop up all of your reads into regions of length k. We were able to relate statistics about which k-mers have mutations in them or not to a model of average nucleotide identity. So just recently, along with collaborators, we're able to look at the number of k-mers that have mutations and derive statistics like variance and expectation that relate this to the actual ANI relatedness between the 2 different genomes. Secondly, there's this new sketching-based technique that you might have heard of called frac-min-hash, which is a way that we can very, very quickly compute statistics about k-mers. And it works basically by chopping up your reads into a bunch of k-mers, subsampling in a very special way, using this list of k-mers as a fingerprint of your data. And then from there, you can get an estimate of similarity, the containment index, that just happens to be, with this formula right here, exactly related to ANI. So from these sketching-based approaches, we can get ANI. From ANI, we can get statistics about what happens to the k-mers. You marry those 2 things up, and that allows YACHT to work. Okay, so how does it actually work in practice? Given the reads in your reference database, the first thing that you're going to do is convert this all into k-mers. Then after that, you're going to run this frac-min-hash subsampling or sketching, denoted here with the red dots. And then we're going to go one by one to all of my reference genomes and say, does it overlap at all with my sample? And if it does, we're going to retain those. And then from that, so we have now reduced our big reference database of like 1 million down to fewer. If you're familiar with MASH, this is like MASH screen kind of thing, but taking just the non-zero hits. So MASH screen sort of thing. And then from there, those unique hits, we are going to get genome-specific k-mers. So give me k-mers that are present in one of these genomes that are not in the other ones. You might be saying, ooh, well, that might not happen if you have a whole collection of strains. Turns out the k-mer size that we're using here is like 60, 90, 120, so massive k-mers. So even if you have very high ANI, you can still have unique k-mers. And then you plug it into our stat formula to get if there is enough evidence in that form of these overlapping k-mers in your sample to represent the claim that this thing is present. So that's basically how this algorithm works. How powerful of a test is it? Well, I told you that we were controlling the false negative rate. And so its power is going to be, well, how is the false positive rate? Unfortunately, to do this, you would have to assume some distribution of A and I values, which is going to be totally different depending on sequencing technology, the environment of what you're sampling, et cetera. So we took a distribution-free approach. And instead, we asked, well, how far apart do the A and I's have to be until our false positive rate and our false negative rate is exactly the same? Because like, if you want the false negative rate set to be 1%, how different do these A and I's have to be until the same thing for my false positives? So instead of talking about the theorem, let me just show you this plot here. On the x-axis, I have A and I. On the y-axis, I have my positive rate. This red vertical dashed line right here is the threshold that I set. Because remember, that's one parameter that you set when you run YACHT. And you say, how big of an A and I does it have to be until I say these two are the same species or genome or whatever? Here, it's set to 0.95. And you can tell, indeed, the false negative rate is controlled. Here it is set to 1%. And if your reference genome is related to at 0.95 or above to something in your sample, this method will correctly say it is present. So over here in this region, it's, ooh, my mouse quit working. There we go. Correctly detected. Over in this region over here, the A and I is big enough, or sorry, farther enough away to correctly reject that genome being present in your sample. So it's this region in here that things can get squishy, and we might incorrectly reject something or incorrectly accept something as being present in the sample because this A and I is in this region. So the power of this test is how large this region is. And you can see that the blue line and this orange line here correspond to the number of exclusive or these unique k-mers that I have. If you look carefully, that's 200 and that's 400. So you notice as we get more unique k-mers, this region, if it was perfect, it would be a perfect little step function going like this. So moral of the story, you don't need very many unique k-mers to be able to get pretty high performance. Great. Okay, what does it look like in actual practice? We're going to use spike in data. I take a real metagenome, a real genome. We mutate reads from that genome. We spike it into my metagenome. And then I try to say, is that thing present or absent, depending on the control over my A and I. Repeat this 29,000 times, and then you're going to get a plot like this. Once again, A and I is on the x-axis. And then here is saying whether YACHT predicted, yes, that thing was in your sample, the spike in was in, or the spike in is not in. Once again, if this was perfect, it would be a nice little step function right here. If it's above the A and I threshold, then you say, yep, it's in. If it's below, you say, no, it's not. And so you can see we have a few different false positives and false negatives here. But if you look at overall the samples, these are very low. If you look at that coverage threshold, which I won't have time to go into too much, but you can basically say that we control false negatives, false positives fall off like expected. On mock communities, this is a CAMI data set compared to one of the most accurate methods, sour mash here. You'll notice that basically we get a massive reduction in false positives with just a slight increase in my false negatives. So concluding with features, future, friends, and funding, there is a preprint up on bioRxiv. It's under review at Bioinformatics. We have some proof of concept code up on GitHub and Python. Basically, this is the first theory-backed statistical test for presence-absence of a genome and a metagenome. Obviates the need for arbitrary thresholding. Please don't threshold anymore. We can actually get, p-values associated with these things and confidences. And we specifically targeted A and I in coverage because these are intuitive things that practitioners can interpret. So in the future, we're going to convert this to a compiled language, so then things will go super fast. That's not super necessary because the Python code will profile something in on the order of minutes, even if your reference database has hundreds of thousands of organisms. I want to thank my co-authors, Steve, Chunyu, and Alexi, as well as funding from the NIH. And thank you for your attention.
Okay, we have time for one question.
I have two questions. I will just ask two quick ones. Does not count. Okay. Yeah, great, David. So two quick questions. First, can you quantify the amount, like doing providing the species in the community? And second, can you extend to genus level or even higher?
Yeah, great question. So yes, you can. The really cool thing about that scaled min-hash estimate is that it works for ANI values all the way down to 50%. So even though some tools like MASH or FastANI or PyANI can't go down that far, scaled min-hash does. And so because of that, you can, if you wanted to, do a genus or a phylum level experiment.